Hello, my name is Griffin Capehart. I'm applying for the zoology position at Santa Rosa Junior College. And today I'm going to give you a talk about what makes an animal. Specifically the origin and evolution of the animal body plan. First of all, we need to determine the characteristics of an animal. In other classes you've talked about plants and fungi and protists and bacteria and archaea. Those animals have their own suite of characteristics. Plants are photosynthetic, eukaryotic, multicellular. Fungi are multicellular, heterotrophic, meaning they digest their food, that eat other organisms. Protists can be both heterotrophic and photosynthetic, or they can't be chemosynthetic, but bacteria can be, meaning they can make food from chemicals without input from the solar energy. Animals are not photosynthetic, nor are they single-celled. So what are animals? Animals, first and foremost, are eukaryotic. Their cells have linear chromosomes that are stored in the nucleus, unlike a prokaryote, and they also have specialized organelles, so their cells are eukaryotic. Additionally, they got lots of them. Animals are multicellular, which makes them different from some fungi, some protists, bacteria, not necessarily plants though. So they're multicellular. All animals have more than one cell. Some only have about 1,000 cells, like the nematodes Caenorhabditis elegans. Others, like humans, have 50 trillion cells. A blue whale may have as many as 100 quadrillion cells. It's a lot of zeros. Finally, animals are heterotrophic, which makes them like a fungus, but we must ingest our food. We don't just spit out digestive juices and digest the food where it is. We put the food into our bodies and digest it inside. So Pac-Man is an animal, assuming he's eukaryotic and multicellular. But he ingests his food. That's what I'm depicting there. Okay, so the origin and evolution of the animal body plan can be broken down into 4 distinct characteristics. First one I want to talk about are the germ layers. And so the germ layers are developmental tissues that give rise to specific tissues in the adult form or juvenile form of an animal. There are typically 3 tissues, although some animals only have 2. The first tissue, though, is the ectoderm. The ectoderm, ecto, outer, gives rise to the skin and the central nerve, not the central nervous system, but the nervous tissue. So the ectoderm, good way to remember that, ecto, outer, skin. Just got to add on to the nervous system to that. So that's the ectoderm. If we have ectoderm, we must also have endoderm. The endoderm, endo, inner, turns into the digestive tract, the lining of the digestive tract, that is, endo-inner. The third germ layer is the mesoderm, or mesoderm. Meso means middle. The mesoderm develops into muscle, bones, cartilage, organs. So animals with only the 2 germ layers don't really have muscles. Those animals are like the comb jellies, the ctenophorans, and they move around with cilia because they don't have muscles to power limbs or anything else. Animals that develop a mesoderm and then develop muscles are very efficient at moving around. And that's why mesoderm is important to the development and evolution of an animal. Next thing I want to talk about is a coelom. So there's 2 different body plans in terms of a coelom. We have acoelomates, the lack of a coelom, and we have coelomates, animals that possess a coelom. So a coelom, what is it? It is a fluid-filled body cavity. That fluid-filled body cavity makes it easier to change the shape of one's body because there's fluid that can be displaced and pushed into other parts of the body. With mesoderm, you can contract your muscle, change the shape of your body cavity, and force that water to a different part, changing the shape of your body as you move, causes movement. Acoelomates don't really have the ability to do that. But the coelom, the development of a coelom, along with that mesoderm, really helped direct the movement of an animal. Okay, we'll come back to the coelom because that'll be important here in a second. And the next thing that we need to talk about are nervous systems. So, some animals have what is called a nerve net. That's just a diffuse network of nerves that covers the entire body equally. Literally like taking a net and wrapping it around an organism. The other is a central nervous system where you have a central governing unit, like a brain. Like here's a ganglia, which is typically what we call a centralized unit of nerves. And so early animals that have only the two germ layers have a nerve net, because they have this body plan where everything is equally likely to get food or move in a certain direction. And so if you take a jellyfish and you rotate it over, you can cut it pretty much any which way, which is called radial symmetry. So these early animals have radial symmetry in nerve nets as their central nervous system. They don't move very much, and when they do move, they're not using a coelom and mesoderm to move their body. They're just kind of moving in whatever direction the nervous system tells them to, and they can move in any direction equally as well. The next body plan with the central nervous system was bilateral symmetry. So you can take this animal, this snake, this rattlesnake, I'm a rattlesnake guy, and you can divide it in half to two equal sides. And this ganglia developed near one end of the body, which led to cephalization, which is the evolution of a head region. And that head region, with that specialized nervous tissue, the ganglia at the very front, made searching and moving and looking for prey more efficient, especially with the coelom and the specialized muscle tissue associated with the mesoderm. So the central nervous system and bilateral symmetry came together. With our bilateral animals, which is pretty much all of our animals besides cnidarians, jellyfish, and ctenophrans, or comb jellies, the only other exception is the adult form of echinoderms, which are sea stars and sea urchins. But with all those other bilaterally symmetrical animals came what we call now are the protostomes and deuterostomes. And bilateral animals either are a protostome or a deuterostome. And the differences are in how they developed. So a protostome, when its germ layers are forming tissues early on, when the endoderm first begins to pinch off towards the center and form what will be the digestive tract, the first pore or hole that develops will become the mouth, will become the head region. So their head butts, their head develops first. In deuterostomes, that pore that first develops becomes their anus. And then the mouth will form. So they're butt heads. So things that are protostomes are things like your arthropods, insects, corals are cnidarians, sorry. Onychophrans, worms. Those are all protostomes. Deuterostomes are the vertebrates, or all the butt heads. The other difference between protostomes and deuterostomes is how the coelomic cavity develops. In a protostome, the mesoderm forms like this, separately from the endoderm. Forms these patches of mesoderm, which then hollow out to form the coelomic cavity. In a deuterostome, the mesoderm forms from outpocketings of the endoderm and then pinch off to form the coelomic cavity. So this is how animals where they've come from and the evolutionary trends that have led to that. Also the relationship between our different trends and the evolutionary traits that those led to as well. That's all I got.